Welcome to the Deep Dive, the place where we take complex biological concepts and show you the surprising genius behind them.
Today we're looking at one of the greatest success stories in all of evolution, the angiosperms, the flowering plants.
And to really get their success, you have to start with a story of deception. I mean, a plant that basically invented the biological dating stamp.
We're talking about the Mediterranean orchid, Ophrys speculum. This plant has evolved this just astonishingly specialized trick.
So specialized, it's only pollinated by one specific insect, the male wasp, Daisiscolia ciliata.
And the flower isn't just pretty, it's a perfect mimic. It visually, tactilely, and chemically dupes the male wasp.
It looks like the female, feels like the female, and even gives off the exact same sex pheromones.
So the male wasp tries to mate with it. Biologists call it pseudocopulation.
And in the process, he just gets a faceful of the orchid's pollen.
It's the perfect demonstration of coevolutionary specificity, and it really sets the stage for our deep dive today.
Exactly. So we're going to tackle the core reproductive biology of these plants, focusing on what some call the 3 Fs.
Right. Flowers, double fertilization, and fruits. We'll map out the whole life cycle, sexual to asexual reproduction.
And then we'll get into how humans are now changing the game with modern biotechnology.
Our goal for you is to take this really intricate process, which is often full of complicated diagrams,
and just distill it into a clear, fascinating story. By the end, you'll know exactly how a flower becomes a seed and how that seed becomes a seedling.
So let's start with the heart of it all, the flower.
The flower itself. This is the specialized reproductive structure anchored to the stem by the receptacle.
And it is an evolutionary marvel built around one thing: efficiency.
Efficiency is definitely the key word. You start with the protective layers, the non-reproductive parts.
The sepals. They're usually small, green, and they cover the bud before it opens.
And then you have the petals, which are basically the plant's billboard, brightly colored to attract just the right kind of attention.
And then we get to the actual reproductive machinery. So the male structures are the stamens.
You can think of the stamen as the pollen factory.
That's a great way to put it. It consists of a stalk, that's the filament, holding up the anther.
And the anther is where the pollen is actually produced inside these little pollen sacs.
Exactly. And on the female side, you have a carpel, or sometimes it's called the pistil.
I always think of this part as the tightly guarded vault.
It is. At the bottom, you have the swollen ovary, which holds the precious ovules, the future seeds.
And then there's the long neck, the style.
And right at the very top, the stigma. It's this sticky landing platform, perfectly engineered to capture any incoming pollen grain.
Okay, so once that pollen lands on the stigma, we've got pollination.
Pollination, yes. The transfer from anther to stigma. But how that transfer happens, that is where coevolution gets incredibly creative.
And it's amazing that only about, 20% of flowering plants actually rely on the wind for that?
Yeah. Things like grasses or temperate trees like hazel, it's a really inefficient strategy.
So they just have to produce huge amounts of pollen to compensate.
Massive amounts. Their flowers are usually small, green. They don't waste any energy on bright colors or nectar. If you have spring allergies, well, you're experiencing that inefficiency firsthand.
But the other 80%, they've mastered biological bribery.
They really have. They use animal pollinators, biotic pollinators, and they attract them with food, usually nectar or pollen.
And the whole flower is designed to make sure the pollinator picks up pollen and delivers it accurately.
Take bees, for instance. They're probably the most important group of pollinators.
They see yellow and blue really well,
Right.
But not red.
Right. And they're guided by scent. Many flowers they visit, like a dandelion, even have nectar guides.
Oh, the UV markings.
Exactly. They're like runway lights that we can't see, pointing the bee directly to the nectar.
That specialization is just incredible. Moths, for instance, they operate at night, so they need something different.
So they prefer white or pale yellow flowers that stand out in the dark. And those flowers are often very fragrant.
Which leads to those really tight, co-evolved relationships, like the yucca plant and its specific moth pollinator.
A true reciprocal exchange. And then you have birds, like hummingbirds.
They're all about pure energy.
Pure energy. They don't have a great sense of smell, so their flowers are usually large, bright red or yellow. But they produce huge amounts of high-energy nectar and have almost no scent.
This reminds me of that famous prediction Darwin made, the Madagascar orchid.
Angraecum sesquipedale, yes. Yeah. It has this massive 28-centimeter long nectar tube.
And Darwin predicted, just based on the flower, that a hawk moth must exist with a proboscis that long to feed on it.
And decades later, it was found. It just shows the power of that evolutionary pressure.
And we're seeing it happen in real time now?
We are, with climate change. In the Rocky Mountains, researchers have found that shorter-tongued bumblebees are being favored.
Because the blooming season is compressed.
Exactly. The tongues of some species have shrunk by about a quarter in just 40 years. That's incredibly fast selective pressure.
Wow. So we've seen the huge scale, the flowers, the long moth tongues, but the real action is happening at the microscopic level.
Inside the ovule and the pollen grain, yes. This is the alternation of generations, but it's super reduced in angiosperms. The gametophytes are tiny and totally dependent.
Let's focus on the results. So inside the ovule, you get the female gametophyte, which is the embryo sac.
And that sac holds the most important cells, the egg cell and two other specialized cells called the polar nuclei.
And those polar nuclei are key for the future food supply.
Crucial. Meanwhile, on the male side, the pollen grain forms. It's really simple. It just has a tube cell
Which will drill a path.
and a generative cell, which will produce the two sperm.
So the pollen lands on the stigma, the tube cell starts growing down the style, making a pollen tube.
And it's guided by chemical signals from the ovule. As it grows, that generative cell divides.
And produces two distinct sperm cells.
Right. And this leads us to the single event that truly defines the angiosperms.
Double fertilization.
Double fertilization. The two sperm cells go their separate ways inside that embryo sac.
Okay, so the first sperm does the obvious job.
Right. Sperm number 1 fertilizes the egg cell. That forms the diploid zygote, which will become the embryo for the next generation.
And the second sperm.
The second sperm fuses with those two polar nuclei in the central cell.
Creating a triploid cell, 3 sets of chromosomes.
Exactly. And that cell divides like crazy to form the endosperm.
This is the real genius of the system, isn't it?
It is. It's pure efficiency. The plant only invests in making that nutrient-rich food supply, the endosperm, after fertilization is confirmed.
So there's no wasted energy on an infertile ovule.
Not at all.
Okay, so fertilization is complete, and now the transformation begins.
The ovule starts developing into the seed, and the ovary that surrounds it matures into the fruit.
And the outer layers of the ovule, they harden up to form the seed coat, right?
They do. It's a tough protective layer. And inside, that zygote starts dividing. But the first division is asymmetrical.
Ah, right. One cell becomes the embryo, the other becomes the suspensor.
The suspensor, it's essentially the umbilical cord. It anchors the embryo and transfers nutrients from the parent plant.
And what about that food storage? The endosperm, it doesn't always stick around, does it?
It depends. In monocots, like corn or rice, the endosperm is the main food source for the seedling.
But in many eudicots, like a bean,
The food gets completely transferred to the seed leaves, the cotyledons, before the seed is even mature. So when you open up a bean, those two big fleshy halves, that's the food storage. The endosperm is already gone.
and all of this leads to dormancy, a critical survival strategy.
Yes, absolutely. Dormancy ensures the seed just waits. It won't germinate until conditions are perfect.
Whether that means a heavy rain for a desert plant or a period of cold for a temperate one.
The resilience is just mind-boggling. I mean, the oldest seed confirmed to have germinated was a 2,000-year-old date palm seed.
Wow, from an archaeological site?
Yes, just incredible.
So when it is finally time to sprout, it all starts with water.
Imbibition, the rapid uptake of water by the dry seed, that cracks the coat.
And the first thing to emerge is the radicle, the embryonic root.
It has to. It needs to anchor the plant and find water immediately.
And the way the shoot gets above ground is so clever. For a bean, a eudicot, it uses a hook.
The hypocotyl, the stem below the cotyledons, forms this tough hook and literally pulls the fragile shoot tip and cotyledons up through the soil.
But monocots like grass or corn do something different.
They use a self-drilling sleeve. It's called the coleoptile.
So it's like a protective tunnel.
Exactly. Coleoptile pushes straight up, and the delicate shoot tip only emerges once it's safely above the surface. It's a marvel of engineering.
Now, switching gears a bit from all this sexual reproduction, plants can also just clone themselves.
Yes, vegetative reproduction, producing genetically identical copies,
And the most common way is fragmentation.
where a piece of a root or a stem just breaks off and regenerates a whole new plant,
And you see this on a massive scale with Pando, that aspen clone in Utah.
which is estimated to be something like 47,000 genetically identical stems, all sharing one massive root system. It's one organism.
Wow. And then there's something called apomixis.
Right, that's the asexual production of seeds. Dandelions do this. There's no fertilization, but you still get a seed.
So you get the dispersal advantage of a seed without the whole cost and risk of sexual reproduction.
Precisely. which really highlights this great evolutionary trade-off.
Asexual is great for rapid growth in a stable environment.
You pass on 100% of a successful genome, but sexual reproduction, that's your engine for genetic variation.
Which is absolutely essential for adapting to change, new diseases, a shifting climate.
Exactly. And of course, you get the seeds and fruits for dispersal.
So because that diversity is so critical, plants have evolved ways to avoid selfing or self-pollinating.
They have. Some have separate male and female plants, or their parts mature at different times. But the most common defense is self-incompatibility, SI.
So this is like the plant's immune system for its own pollen?
That's a perfect analogy. It relies on specific genes called S genes. If the S allele in the pollen matches an S allele in the stigma,
The plant rejects it.
it rejects it. The stigma can release enzymes that just block the pollen tube from growing or even destroy its RNA.
It's fascinating. Nature builds these incredibly complex systems to control reproduction and ensure diversity.
And now humans are stepping in and, well, intentionally overriding those rules through biotechnology.
Of course, we've been modifying plants for thousands of years.
Absolutely. Traditional breeding, or artificial selection, is how we got modern corn from a wild grass called teosinte. Breeders just hybridize related species to select for traits we want.
But modern genetic engineering is on another level in terms of speed and specificity.
It really is. Biotechnology lets us transfer a specific gene, the transgene, from a completely unrelated organism. It bypasses the natural limits of hybridization.
And that speed is why so many argue it's essential for solving our immediate global challenges.
Especially food security. With finite land and water, we need to increase yields.
And one of the biggest applications is pest-resistant crops, bee crops.
Right, using genes from the bacterium Bacillus thuringiensis, these crops produce a beta toxin that's harmless to us.
And that's because it's a protoxin, right? It only becomes active in certain conditions.
Exactly. It only activates in the high alkaline environment of an insect's gut. Our stomachs are acidic, so it just breaks down harmlessly.
And there are these layered benefits, too, because bleak corn has less insect damage.
It's 90% less likely to be contaminated with pseumonicin.
A really nasty fungal toxin that's a known carcinogen.
So it's a huge public health benefit that comes directly from the pest resistance.
We're also using this for biofortification, improving nutrition.
The classic example is golden rice. It was engineered to produce beta-carotene, a precursor to vitamin A, directly in the grain.
And this is a targeted effort to combat childhood blindness in parts of the developing world.
Right, and the scope even extends to energy, with biofuels.
Using plant biomass like switchgrass to create fuel.
Yes, the idea is to create a carbon-neutral energy alternative, since the crops reabsorb the CO2 they release when burned.
Of course, whenever you deploy a technology on this scale, there's going to be controversy.
And the main concerns have always been ecological, the unforeseen risks.
One of the first big stories was about blight corn pollen harming monarch butterfly larvae.
Yes, that was a highly publicized lab study, but the follow-up field studies largely refuted those initial claims.
They found that in the real world, the concentration of toxin on milkweed leaves was negligible.
And more importantly, that traditional chemical pesticides were actually far more damaging to monarch populations than the wheat pollen was.
The more serious and I think more enduring concern is transgene escape.
Definitely. This is the risk of a transgene moving from a crop into a wild relative, creating a potential superweed.
Like a weed that's suddenly resistant to a broad-spectrum herbicide.
And this isn't hypothetical. It happened in Oregon back in 2003 with glyphosate-resistant creeping bentgrass.
It escaped from experimental plots.
By wind. And just a few years later, over 60% of the wild grasses nearby had also become glyphosate-resistant. It shows how hard it is to contain these genes once they're out.
But scientists are actively working on ways to prevent this.
They are. Clever solutions, like engineering male sterility into the transgenic crops, or an even more sophisticated approach.
Inserting the gene into the chloroplast DNA.
Exactly. In most plants, chloroplast DNA is only inherited from the egg parent, so it can't be transferred by pollen at all.
So we've dissected this incredible sophistication of angiosperm life.
From the efficiency of the flower to the biological trick of double fertilization, the resilience of dormancy, and the trade-offs between sexual and asexual life.
And finally, looking at the immense power we now have to accelerate evolution through biotechnology.
Addressing really urgent needs like nutrition and pest resistance.
It's the ultimate tension between timescales, isn't it? We started with that slow, precise co-evolution that took millennia to produce an orchid's disguise and a moth's long proboscis.
And now we're trying to achieve similar precision, to design the perfect plant, but at the speed of a lab bench.
Solving problems in a single growing season. So we'll leave you with this final, very complex question for your own deep dive.
Given the verifiable power of plant biotechnology to quickly solve immediate human problems like malnutrition, should our ethical priority be maximizing that short-term benefit?
Or should we slow down this biological experiment to guarantee safety against hypothetical long-term ecological risks like transgene escape?